In March this year, 3 weeks ago, we hosted the 6th International Multiple System Atrophy Congress. This was a scientific meeting for basic scientists, clinical researchers, advocates, industry, and academic people to come together to share their ideas and to talk about progress in multiple system atrophy and really where we need to go forward for the future. Now, there are several reasons why I think you should feel very encouraged. MSA is now getting the attention that it deserves. Over 250 people from all over the world came together to speak about this rare disease. And we had 70 young investigators from the world's best laboratories and clinical research centers that came to present their work. And for a rare disease, this is really an excellent turnout. We invited 44 leaders of the field to come and speak about multiple system atrophy. And anybody that sees this list of names here, you'll recognize that these are some of the giants in the field of neurology, of movement disorders, and of sleep disorders. And we were very excited to have them with us. We asked these people to share their progress and their ideas, but also challenge the findings of others and the applicability that may have to MSA patients, and question one another's approaches. So the goal here really was to have a lively discussion about some of these advances. So in the program, we covered a number of different advances. We spoke on the first day about the biology of MSA, and on the second day, we spoke about research, treatment, and diagnostic criteria. And I hope over the course of the next 20 minutes or so, I'm going to give you an idea of why we chose those topics, why we put them together, and why that they may be relevant to you. Now, the plenary lecture was given by Dr. Stanley Prusiner, who is a world-famous scientist known for winning the Nobel Prize in 1997 in physiology and medicine. And this was for the revolutionary concept of proteins as infectious agents. And this is something that is undisputed today, and it truly illustrates what's at the core of the disease process in multiple system atrophy. So here's Dr. Prusiner giving his plenary lecture, and we're very fortunate that in recent years he's begun focusing on multiple system atrophy in his laboratory. In 2013, the Prusiner laboratory produced really a groundbreaking study that showed if you take the MSA protein and you inject it into a mouse that is genetically engineered to be susceptible to neurodegeneration, that mouse has a rapid decline and within 100 days develops neurological signs that sort of mimic what we believe MSA would be in a rodent model. And if you look at the brains of those mouse that were infected with the MSA protein, they show exactly the same pathological changes that you see in multiple system atrophy. Just to go on from this concept a little bit, of course, the alpha-synuclein you've probably heard quite a bit about over the course of this morning. This is the rogue protein that's really at the heart of MSA. It's abnormal forms of these proteins that clump together within the cells of the central nervous system and the spinal cord, and they injure those cells, and they cause their destruction. And these same clumps are found in patients with Parkinson's disease. So, of course, alpha-synuclein is a protein that is normally produced in the body. And like every protein, it continuously lives in a state of being unfolded and folded. And when it folds, as I'm illustrating here with this paper airplane, it forms a final shape with a biological function. Now, alpha-synuclein can also be prone to misfolding. So it can misfold into an abnormal conformation, which it exists in a different form. And what that abnormal form of alpha-synuclein can do is it can interact with other synuclein proteins, and it can cause them to also adopt this misfolded conformation, and this that clumps together and injures the cell. And these abnormal proteins can spread from one cell to another, which is the very concept of proteins as infectious agents. Now, of course, the work of Dr. Prusiner is really fascinating because he's been working on prion disorders. These are what we call prion proteins for quite some time. And he shows that if you destroy the protein, the infectivity goes away. So this raises the possibility of stopping the spread of synuclein to other cells. So I want to take the bull by the horns a little bit when it comes to this prion hypothesis, because for patients, it is a controversial issue. This is not an infection like a virus or a bacterial infection. This is the concept that proteins can infect cells, and it's the normal protein within the body that spreads from one cell to the other. There is absolutely no evidence that we have that MSA is transmittable to humans. And I'm referring you to this recent editorial that just came out in the Annals of Neurology by some of the leaders in the field of MSA, including Gregor Wenning, Dr. Kaufmann, etc., etc. And they reviewed this in detail. So we've no evidence that it's transmitted from one person to another, even in close contacts from the same household. So caregivers are not affected once they've been caring for a partner with MSA. And there's no data to support that the protein can be spread through things like blood transfusions or organ transplants. So when you hear this concept of prions, don't think of it as something as infectious per se, like we understand as a virus. It's a concept that the protein itself can infect other cells. I just want to put your mind at rest about that, and I really thought it was important to talk about. So we moved on from the concept of prions and the prion hypothesis to begin talking about disease-modifying strategies. Now, when it comes to disease-modifying strategies, as you heard this morning, these are treatments that act at the core of the disease, act at the cause of the disease, and they may have side effects. I mean, think of it like cancer therapy or something like that. You may lose your hair. I'm not saying that this concept would happen with MSA treatment, but when it comes to developing disease-modifying treatments, you want to halt the course of the disease, and you may have to have more tolerance for side effects. So they may not necessarily make you feel better, but they will prolong, with the hope is that they will prolong survival. So we started the conference right after the plenary lecture with a talk on new treatments that are in the pipeline. And here I'm showing you Patrick Brunding, who was one of the chairs of that session. Now, the real conclusion for that from that session, which is why I think it's important to talk to you about this today, is that we believe there's now really a time for hope. The insights into the insights and our new knowledge mean that we're truly learning what the disease is about. And this is something that we can build upon to develop a therapy that can specifically target MSA. Now, when it comes to approaches to therapy, we need different approaches. Of course, we need to stop the spread. But for those that have had the disease for a long time, we also need ways to restore function. So this is why when it comes to therapies, we need everybody's minds thinking together and coming up with different strategies, because there are different things we need to do at different stages of the disease course. Now, one possible approach that was discussed, which I think is very exciting, is the possibility of using vaccines for multiple system atrophy. And here I'm showing you a picture of NYU professor Dr. Thomas Wisniewski, who has done seminal work on vaccines in prion disorders that affect deer. So what can we learn from deer when it comes to MSA? Well, deer in North America and elsewhere have chronic wasting disease, which is known to be a prion disorder. Remember this infectious protein. Now, we can immunize deer, we can manipulate their immune system, and we can target these infectious proteins and protect the animals at risk. So we might be able to adopt the same approach for multiple system atrophy, and that's something that we're currently researching. We have a new project funded by the MSA Coalition to work with Dr. Wisniewski to look at the composition of proteins in the brains of patients with MSA. Because we believe that if we can better understand how these normal clumps come together, how we can recognize them, we could then maybe stimulate the immune system to either prevent them from forming or clear them out. So when it comes to new therapies in the pipeline, there was really quite a lively discussion on this. We spoke about, here you see the Artemis Consortium from Europe. They also presented their work on different strategies. We spoke about various different things. We spoke about vaccines to protect those at risk in the population. We spoke about ways of preventing the formation of alpha-synuclein. We spoke about ways for, if those clumps are formed in the cells, how we can clear them out and restore function to the cells. We spoke about things like stem cell therapies and the possibility that we could promote cell growth or increase cell survival using those techniques. So there was really a number of different strategies that were considered and that were hotly discussed. So we moved on really to talk next about the biology of multiple system atrophy. And one of the things that we want to understand better is there is a specific type of cell that is affected in multiple system atrophy. And those cells are known as oligodendroglial cells, which I'm showing you here in blue. And you can see that each of those oligodendroglial cells has sort of projections where it wraps around nerve axons. And nerve axons are really the pathways. They're really the connections by how one nerve speaks to the other. And what these oligodendroglial cells do is they wrap around the cells, the nerve axons, they insulate them, and they provide support to each of the cells. And each oligodendroglial cell supports multiple different nerves. So as you can imagine, when you affect one oligodendroglial cell, you affect downstream multiple different neurons. That's why MSA is such a rapidly progressive disease, at least in the best of our understanding. So of course, when you see these cells taking up these clumps of alpha-synuclein, you're going to have a drastic effect on the neurons that they provide support to. So for any clinical trial of disease-modifying strategies, of ways to halt the progressive degeneration of the cells, to be successful, we need ways to track changes over time. And we have to have what we call the best outcome measures, which are things that we can measure to show whether a disease is working with the best possible chance and to do it in the quickest possible time. So the things that were discussed in the first day were the possibility of using imaging and specific brain MRIs, the possibility of using a blood test to be able to look at different levels of signals from that could signify the disease state, as well as the other possibility of other bodily fluids like cerebrospinal fluid. So when it comes to the progress we've made in clinical trials for multiple system atrophy, we don't yet have a drug that can stop the progression of MSA. The clinical trials that we've done so far have led to a better understanding. We're learning which approaches make sense and how best to measure what we call efficacy, how well a drug is working in patients. And we're applying the knowledge to what we've learned to plan clinical trials so that we have the best chance of succeeding for the future. Now, I'm putting this slide up here because this was the end of the treatment, of the disease-modifying strategies treatment session. And after each of the talks was given, there was a chance for the audience to stand up and ask questions. And you can see here, I just want to put this up to illustrate what a lively discussion this was, how excited people were and how passionately they spoke. They interchanged ideas, and they really, because the science of MSA will advance faster if we question as a group whether we're on the right track or not. So it's a good thing that these people were passionate, arguing, and something good will come out of this. So the next thing we moved on to talk about was the genetics of multiple system atrophy, which is another thing that I think is really important to talk to you about today. To the best of our knowledge, MSA is a sporadic disease. Aside from some very rare cases in Japan, there's no evidence of hereditability in families. But if we can find certain genes, if we can find what's in somebody's DNA that makes them susceptible to getting multiple system atrophy, it gives us a potential window into the very beginnings of the disease process. We might, in the future, be able to look at somebody's genome and decide and pick out their particular risk because they have particular genes that may make them more susceptible to this. So this is really an exciting field that's moving forward in MSA. Now, in order for that to work, we must work together, right? I mean, this is something we've echoed as a community. Patients know it very well, and the research in the clinical community are also starting to come together. So we have a natural history study where we focus majorly on multiple system atrophy. And we held an investigator meeting. And one of the things that was really exciting about that, we have 22 sites throughout the world, in Europe, in North America, in South America, and in Asia, where we are working together. We're all collecting our clinical information from patients with MSA. We're contributing it to a big database, and in that, we're using that information to track MSA. So we can know what are the key features that decline over time, how fast they change, what treatments influence outcomes, et cetera, et cetera. So I just want to emphasize the point that we're we are working together as a research community on this, and this is a project that's supported jointly by the NIH and the Multiple System Atrophy Coalition. Now, we must work together, but there's several ways in which you can help. You can give your what we call biospecimens to be tested, your things like your blood, your saliva, so we can look at your genome, your genetic makeup, and also the analysis of your proteins in your blood or saliva, or indeed any other body fluid. So I would encourage you, if this is something that you can really do as a community, to move the field forward. The second day of the Multiple System Atrophy Congress was devoted to symptomatic treatments. Now, different to disease-modifying strategies, symptomatic treatments are drugs or devices that you can use that will improve a particular symptom. They will make you feel better, but they won't necessarily alter the course of the disease. So it's clear for you to understand when you're participating in a clinical trial, am I participating in a disease-modifying trial where I potentially am affecting long-term outcomes, or am I participating in a symptomatic treatment which is going to make one particular symptom feel better? Symptomatic treatments are important because they can improve a patient's quality of life, of course. So we discussed various different symptomatic treatments, from new treatments that are emerging to treat the blood pressure, both the high blood pressure and the low blood pressure. We discussed the best ways to treat the sleep abnormalities in multiple system atrophy that include this REM behavior disorder, acting out dreams at night, and sleep apnea. Also, how to manage the breathing issues that occur in multiple system atrophy, like respiratory insufficiency or stridor, that high-pitched noise that you get when breathing. We discussed also cognition and psychiatric problems in patients with multiple system atrophy. We know, and we're increasingly recognizing, how common depression is in patients with MSA, and what we can potentially do to help mitigate those symptoms. So that's something, typically, like while dementia is not typically a feature of multiple system atrophy and would point to another diagnosis, we are starting to recognize that there might be some cognitive and psychiatric issues that come along. We also discussed the various strategies for helping bladder dysfunction and the different option of drugs that we may have and potential therapies that might be on the horizon for that. And again, we discussed how to help with the difficulty moving. But the main conclusion from that session was that a team approach is the best way to manage the symptoms of multiple system atrophy. You may have a sleep doctor that helps you with one part. You may have an autonomic doctor that helps you with your blood pressure. You may have a urologist that helps you with your bladder issues. It takes a team, okay? A multidisciplinary team, and one doctor may not have all the answers. You may need multiple different specialists. So, in order for new treatments to be tested properly, we need you, okay? We really need you to participate in clinical trials, and in good clinical trials where there's clear endpoints, well-designed studies. One great source of information that is available is on clinicaltrials.gov, which is a website. I'll make sure you all have this information, but it is a website that provides you the details of a trial, whether you meet here you can learn what's required from the trial, whether you meet the enrollment criteria, how many visits are needed, how many tests that you'll have to go, etc., etc. So that's really a place where there's a wealth of information. And, to spread the word, if you don't meet the eligibility criteria for a particular research study, tell somebody else in the community. They may well meet those criteria. In order for us to bring treatments to the clinic, because MSA is such a rare disease, we need your help in participating in spreading the word about trials and getting the message out there. So the rest of the session on the second day was really focused on the diagnosis of MSA and the clinical criteria that we use to recognize patients with MSA in various stages of the disease. And what's really clear to us as a community is we need to become better at diagnosing MSA earlier. I'm showing you this little graph just to illustrate, there is this what we call early stage disease, and we're recognizing this increasingly now. This is where the symptoms of MSA are emerging. Maybe symptoms with the bladder, maybe symptoms with the sleep abnormalities, etc., etc. But the person doesn't meet what we call the clinical criteria to match the full MSA diagnosis. But this is definitely this window of opportunity where if we can recognize MSA earlier, we can intervene quicker when we do have disease-modifying strategies. So on average, what we're looking at is it takes around 3.5 years, sometimes close to 4, after a person's first symptoms emerge to when they actually receive the diagnosis of MSA. So as a community, we're trying to come together to figure out how we can do this faster. So when we have the global MSA registry that you all know of, and I'm going to speak a little bit more about that later. But when we did a survey late last year in which we reached nearly 400 patients with multiple system atrophy, and we asked them a bunch of questions so we could get their ideas, we could get their voice, we could get their feedback on what were their symptoms, what were the things that mattered. And one of the first things we asked patients was, what was your first symptom? What was the first thing that you recognized before you got the diagnosis of MSA? Again, we wanted to see what we should be looking for as a community. And you'll see in the bar graph here that 50% of the people that we surveyed said the first problem they had was with difficulty moving, be it walking, clumsiness, or falling. Then around 1/3 of patients said they had blood pressure or bladder abnormalities. A smaller percentage said they began with acting out dreams, this REM behavior disorder, and around 7% said they were unsure or they couldn't recall or had other issues like breathing difficulties. All of these features are well described in multiple system atrophy. I mean, we recognize this as part of the clinical constellation that makes up multiple system atrophy. But none of them alone can actually predict that a person has MSA. But it's when you start combining them that they start to have what we call predictive value. If you have a patient in front of you and it has a combination of various different factors, we should be having red lights come on in our head and saying, hey, maybe this person is at risk of developing MSA. But we need to be very specific when we do that. So we looked at the published literature, and this is something that I was very involved with in part of the MSA Congress about the early diagnosis of MSA. And what we did is we looked at all the studies that had been published that addressed early symptoms or early phase disease, looking back. So when a patient comes to the clinic and you ask them, which is what's called a retrospective study. And we also looked at what were the constellation of symptoms that predicted MSA in studies that recruited patients and watched whether they emerged with MSA, which are called prospective studies. So in the retrospective studies, it was very interesting that the onset, meaning the average age, this is the average, right? Some people have early onset MSA, some people have late onset MSA, but when you put them together, on average, the age of onset is around 56 years of age. Around up to 75% of patients, the first thing that they complained of was difficulty moving. This could include like walking problems, slurred speech, or clumsiness, but the vast majority of people that we surveyed, and as well as those published in the literature, say that they began with subtle abnormalities moving. Around 20%, 28% began with autonomic symptoms. Remember, the autonomic nervous system is the part of the nervous system that controls all the involuntary actions that we don't think about, things like breathing, blood pressure. And if you look at autonomic symptoms from autonomic clinics, as expected, the percentage of people that began with autonomic symptoms is much higher when you're looking at it from an autonomic clinic perspective. The vast majority, 16% of people began with difficulties with the control of the bladder, either urine voiding difficulties, urgency, retention, or incontinence. It's interesting to note that 58% of the men said that even before the bladder problems emerged, they were having problems with erectile dysfunction. And then we have 11% that began with orthostatic hypotension, dizziness, this fainting or lightheadedness when standing. Then we had around 2 to 4% of patients that said they began with respiratory symptoms like stridor or respiratory insufficiency. So again, this is a rare, what we call atypical presentation of MSA, but can happen and can be the story of how the first feature that certain people had. there weren't any real good retrospective studies looking at the start of MSA in terms of REM sleep behavior disorder, but there have been excellent prospective studies by Dr. Postuma and his collaborators that show that around 5% of patients that are seen in a with REM sleep behavior disorder and followed very closely may actually turn out to have multiple system atrophy. So why I'm telling you this is because if we can start recognizing this cluster of symptoms, if we can start thinking about this may possibly mean MSA, we can combine these features. We can have our urologists looking for blood pressure issues. We can have our blood pressure clinics looking for REM sleep behavior disorder, and we have more chance of flagging patients with MSA in the very early stages of disease. And hopefully we can provide faster diagnosis this way. We want to be able to reduce the time from 3.5 years to get that sooner. So we had a roundtable discussion about all of these issues where we brought experts in the field. You see we have Philip Lowe, Edouard Tolosa, Dr. Kaufmann, Dr. Poe. We have the team from Europe and Australia. So we had multiple different continents represented. We had a very lively discussion about whether or not we can really do this, what are the steps we need to do for the future. And basically, again, it comes down to the same issue. We need to collaborate. We need to find those patients. We need to follow them. We need to come up with maybe a blood test or another set of a clinical test, maybe an imaging test that can give us certainty that we find a patient with MSA that's at risk and we can follow that patient and we can intervene sooner with better treatments. So I want to end with just a couple of highlights again. You know, in order for us to really take MSA further, we must inspire the next generation of physician scientists to take on the disease. And we were delighted that at the Congress, we had 70 young investigators from fantastic laboratories from all over the world that came with the opportunity to learn about MSA and how to treat the disease. We gave away 20 travel stipends because we got an R13 grant award from the National Institutes of Health. So we were able to support 20 young people to come to the meeting, to present their work, to really learn about MSA from the biology to the bedside. So that was, you know, we are there, we are making sure young people are exposed, we're trying to keep them in the field, and we're supporting them on their research work going forward. So these are the travel stipend awardees. I'm not going to read you the list of names, but you can know that they were from South America, North America, and Europe also. So finally, I want to kind of pull this together for you, and I've given you a lot of information. I've told you that, you know, we're moving ahead fast. We don't yet have a cure, but we are really getting there. We're thinking all together. We're coming up with different strategies, and something will emerge. There's really a time for hope. There's really progress being made, and hang in there with us. But there are certain things that you can do. Okay, you can participate in research that advances our understanding of the disease. Things like brain scans or lending your clinical information to an observational study or helping us better understand the disease. You can join the global MSA registry to share your voice. My assistant here today is the youngest fellow in our center, and I hope one day he's going to be the recipient of an R13 Travel Award, and he has some information as to how you can join the Global MSA Registry, so he will be handing those out. You can also join the natural history study, which involves you being seen at participating clinical sites that and lend your information, and that information will become part of our database and will help us understand the disease better. Like I said, you can give your biospecimens, things like your blood, your saliva, skin biopsies. Those kinds of pieces of information are very vital to us. And I want to touch on one thing that is never an easy topic, but brain donation is an incredibly important thing that you can do to help advance our knowledge of the disease. It is a difficult process to set up, and we would urge you to have this conversation. If you are willing to do it, to be prepared for us. You have the number. We are looking for brains to help understand better the causes of the disease, to be able to recognize the protein clumps, to be able to find strategies that can pick them up, clean them out. And for that, we need your help. we'll be willing to provide the name of our clinical coordinator that can help facilitate that process. But I would urge you to consider this because it is really a way that you can really help advance the field quickly. So I want to leave you on that note with reasons to be encouraged. Over the past 2 years, we've made real progress when it comes to multiple system atrophy. We're learning about the biology of MSA, and that's helping us develop new therapies. We're closer to diagnosing MSA sooner and very accurately, and we can measure the progression of the disease at various different stages so that we can test potential new therapies that will work, and we're all working together. So finally, I want to just acknowledge that, this meeting wasn't possible to bring all these people if it hadn't been the support of some really fantastic people who helped pull it together. We had the support of not only the academics that came to the meeting, but also the National Institutes of Health, pharma companies, industry like Lundbeck and Theravance, who helped dramatically, and as well as professional societies like the Movement Disorder Society, and the support of advocates like the MSA Coalition. So finally, I just want to end by thanking my amazing team at NYU who worked on this project with us for 2 years, Dr. Kaufmann, Dr. Palmer, Dr. Jose Martinez, our nurse practitioners, Lian Lug, Vivian Cho, and Joy Wong, who without their help, this would not have happened. My collaborators in the Autonomic Disorders Consortium, who we work together on the natural history study, and my research support. Thank you very much.
This is going to sound like a little bit of a ridiculous situation, but about a year ago, my sister and I, we found this study that was done on mice. That I guess they did a study that said that they tested insulin in mice and they had some positive results. So it just so happens a friend of ours, a really good friend of ours, is a pretty prominent endocrinologist. And we asked him if there would be any kind of like drawbacks to trying insulin on my mother who has MSA. So we tried it. We actually gave her twice a day. What was it? 5? It was 10 milligrams. We injected her twice a day with 10 milligrams. Just based on this mouse study. I mean, we just said, hey, it's a shot in the dark. Maybe it'll work. And we did it for like 3 months. And then she wasn't reacting well to it. She was losing a lot of weight. She couldn't eat on the insulin. She was very unhappy. And we had no idea to know if it was working or not. So we stopped it.
Right.
But yeah, I didn't hear anybody mention that. But did you know of anything about this insulin?
And I think you bring up a really valuable point. I mean, one thing is mice and one thing is humans, right? And we can do a lot of what we call preclinical testing before we take drugs or compounds or strategies into patients by looking at animal models. And many times you genetically alter those animal models to make them produce more synuclein protein that can make it look like MSA, or you can produce toxic lesions or something. I mean, there are multiple different things you can do with the mice that supposedly gives you the mice version of multiple system atrophy. We can test potential strategies for treating the disease in those mice, but we don't know if that translates to humans, right? So what we have to be very careful with is taking evidence of this was found in a mouse, it was incredibly successful in a mouse, and thinking that it is going to work in a patient with MSA. And insulin, it's not a benign thing to inject, right? I mean, and you have to consider also side effects. Of course, we all want a cure, but we want something that is going to work and not hurt the patient tremendously badly at the end of the day. So I would just urge caution. When you read those things, it's incredibly exciting, the work that's been done. Those animal models can be so useful to help us understand the disease, to come up with like a test bed for new therapies. But consider twice, just thinking it can directly translate to the case of your mom.
Absolutely. But I guess the question is, did you hear of anything with insulin or any studies that...
We reviewed all the clinical trials that have been done so far, and that was done in the clinical trials session. And so far, we haven't found a clinical trial that has a positive endpoint for a disease-modifying strategy. We have positive endpoints for symptomatic treatments, but not disease-modifying at this point.
Is there a drug that's in clinical trials right now that shows the most promise? Like, I know there's some that are in, like, there's one, this Afaris drug. I don't know how to pronounce it, right? That's a phase 1 clinical trial.
Yeah. Phase 1 clinical trials, there are different stages of,
Right.
of clinical trials,
Yeah.
right? So phase 1 is where you give it into healthy people and you make
Safety,
sure it
right.
doesn't have like terrible side effects.
Right.
Now then it will go into what the target population, which would be MSA patients in a small controlled study to see if it's safe and there are any potential signs that it could be
Right.
helping. That's called a phase 2 study.
Right.
And then it will go into a larger population of MSA patients in a phase 3 study.
Right.
So it's important to consider when you're in a phase, and at each phase, drugs drop out, right? You may have
Right.
great candidates and they drop out. So something that is in a very early phase 1 trial isn't likely going to make it to the clinic until it's gone through,
At least phase
till it's
3.
past
Right.
that, till it's
Right.
been shown to be safe, and then
Right.
progresses onwards.
Right.
There was a study done by AstraZeneca that on an MPO inhibitor that they had recently completed the trial. Unfortunately, it didn't have the outcome that we hoped that it would have. But again, we learned a lot from that trial, including what are the best endpoints to measure. I mean, when you're dealing with a disease as rare as MSA is, it's not diabetes or it's not hypertension where you can have like thousands and thousands of people in a trial. So the fewer people you have in the trial, you need to have stronger what we call outcome measures so we can cope with that what we call low statistical power.
Yeah, no, I see the challenge. So what's the latest phase that you see right now? I mean, has any drug made it past phase 2 for MSA right now?
Not at present.
Okay.
Not at present. There's an open label. We spoke today earlier about stem cells, right? I mean, there's a very small controlled study that has been done by the terrific team at Mayo Clinic that are really testing the possibility of stem cells, and it's not an official phase 3 trial, but they're able to keep patients with MSA on stem cells to see how they will evolve over time. So there are things in the pipeline. There are trials that are going on. There are things that are not working. But even if a drug doesn't work, it doesn't mean that we're not learning how better to do it for the future, whether that strategy makes sense, whether it doesn't, should we reconsider, should we combine it with something else, et cetera, et cetera.
Got it. Thanks for all your work. Thank you.
Far enough for a pharmaceutical or the researchers to say that?
I mean, it depends on the outcomes. I mean, companies will take things forward if there's a very strong sign in phase 2 that it is going to work, right? So right now they're kind of pulling the data together, discussing different options, and we are just waiting to kind of find out what's going to make it past phase 2. But again, drugs drop out at different stages. So if you're in phase 3 clinical trials, you're more likely that drug is going to make it in the end than possibly something that's in phase 2. But there's still no guarantee.
Question on insulin in MSA. I do know there's been a human trial in exenatide in Parkinson's patients with the idea of slowing down the progression of Parkinson's. This was work done by Parkinson's UK. I've been told that the original data was very positive, but the second follow-up was not as positive. So I think to answer your question, there's still a lot of work that needs to be done in what is these repurposed drugs.
Well, quickly, 2 questions to you. Is there anything more on the ocular or the retina scanning for how long people had MSA or?
Yeah, we're currently funded by the Michael J. Fox Foundation to look at the retina. But the retina has a lot of nerves. It's like an extension of the brain, but you can image it, and you can get these high resolution pictures that are at higher resolution than you would typically see in an MRI scan. So one of the things that we believe is the retina could be a very good way of measuring nerve degeneration. And all you do is you sit, you look at, you fixate on a dot, and you have sort of an ultrasound scan that can reconstruct the layers of the retina. So with the Michael J. Fox Foundation, we are following the retina over time, prospectively going forward, in patients with Parkinson's disease, in those at risk of getting either MSA or Parkinson's disease, as well as a cohort of patients with MSA. So what we're going to be able to do is look at the decline in the retina, hopefully over time, and we're going to know how patients behave outside of a clinical trial, how fast the retinal nerve fiber layer thins. And then hopefully our idea is that if we know that it decreases by a certain amount in what we call the natural history of the normal progression of the disease, we can then compare, if we treat with a drug, can we slow down that thinning? Could that be a marker of less degeneration in a particular region? So we're actively working on that. Yeah.
My other question was, I was watching something on Parkinson's, and they said alpha-synuclein started in the gut and spread into the brain. Is it the same for MSA, or is it?
That is a theory. I mean, and it's based on, one of the high risk factors for Parkinson's disease is constipation. And you see, if you put together large population-based studies of epidemiological data, you'll see that patients with constipation have a higher risk of developing Parkinson's disease. And of course, the gut is very densely innervated by nerves. So the idea is that maybe these nerves can take up synuclein, and it can be like transported to the brain and then pass from one cell to another. That's in Parkinson's disease. We don't know in MSA. I mean, MSA affects predominantly a different cell type. In Parkinson's disease, you get these abnormal clumps of synuclein in the nerves, right? Whereas in multiple system atrophy, for reasons we don't know, it mostly affects these glia cells that, they spread and they support multiple different nerves. So how it gets there, why it's taken up by those cells, we still don't know. But the microbiome and areas like that are hot topics now for research in MSA too. So hopefully we'll have a better understanding of that in the future.
It's around what was learned at the MSA conference or convention that you went to, and it sounds like education was a big part of it, and I think education is a big part of understanding what this disease is and what it does to you. I found that to be true right there in Princeton, and I went there several times over several years, and they finally got the point. They got the point that education is, with me, it sounds like we don't have enough education. And so my question to you is, what do we have to do to spread the word? What are you doing specifically to spread the word? And I know we are the greatest advocates for it because I find myself with it.
I think you're absolutely right that education is foremost, which is one of the reasons, we wanted this meeting to cover not sort of experimental things, if you like, but really to provide like a state-of-the-art summary of where that particular field of MSA research was right now. So it's fundamentally important to educate people. I mean, I was listening to the stories this morning of the lady that was saying, well, I don't live close to a specialist center. My doctor doesn't know what MSA is. We and other research groups publish extensively on MSA. We publish articles that are, expert reviews of how to treat the autonomic problems. There is a European and a US task force that met in Bologna early last year to put together guidelines for the treatment of stridor and respiratory abnormalities to sleep apnea. So I think that if we can get that information out, one thing that you can do is get that information into the hands of your physician. So I would like to work with all of you in having, because many times we deal with rare diseases, right? That's what we do at the Dysautonomia Center. Many times when you're doing that, you're relying on the local physician to be the eyes and ears on the ground. So if you can talk physician to physician, almost by giving them, very good expert guidelines, a current review of the literature, that person can read that article and can know the background, the relevant treatments, the things to look out for, the side effects. And that's one way I think that you can help to educate your physicians in that way. You can always tell your physicians, look, there's this expert center for MSA at NYU. This is, you have on the back of there, you have a phone number. I mean, if your physician is struggling with some of the complications of the disease and wants to have a conversation with us, we would be more than happy.
what exactly does it mean? The entire brain is donated, or just a part, or just samples? Do you know what that entails?
The entire brain. And then you can consent for, if you want to give other pieces of tissue, you would consent for a whole body autopsy where they would take samples of different regions. So typically the whole brain is taken. It is one of the reasons we kind of urge people to discuss this with us and to kind of put it in motion is that you have to prepare those samples in a particular way to make them be usable, right? Or to get the most out of that tissue because it is so very valuable. So if you know, we would encourage you to, you know, call us. We can help you set up the paperwork to do that. We can facilitate the process a little bit, and we can also help to make sure that when it happens, the sad situation of losing somebody, we make sure we get that tissue in the best possible way to use it.
Is it true that there's also a time constraint? You have to harvest the brains within 24 hours or 48 hours of passing away?
It's very ideal because remember that the tissue is fragile, and that if you have too long a delay, you lose the quality of the sample. So, you know, I mean, these are things that if you are considering that, let us make sure that we have the right pieces of information in place so that we can get the most, we can learn what we need to learn from that tissue.
My question is kind of twofold. Number one is, how do we, meaning kind of the Northeast area of America, compared to other parts of the country in the incidence of MSA compared to, Europe?
It's a good question. I don't know. It's a good question. I mean, I do. There are two forms of multiple system atrophy. There's the Parkinsonian type and there's the cerebellar type. In the Japanese cohorts, the cerebellar type is more common, whereas it's not more common in the Northern American cohorts. So there does seem to be some population differences. Probably, maybe I'm speculating here, but could have something to do with the genetic background of somebody. But again, you know, I really don't know that question. It's a very good question.
Okay, so my second question is that in 1993, my dad passed away at 59 years old from a neurological disease. We live in, again, way out there, small town, and we were told by his neurologist that he died of ALS because there's no other neurological disease that kills you. Since the time that I started having symptoms, he had an autopsy. They saved slides and parts of his brain that then went on to Mayo Clinic in Jacksonville. We sent it there because when I got seen for Mayo Clinic in whatever state, Minnesota, it starts with an M or something. Jacksonville called me and wanted my brain, and I said, "Well, I'm still using it, but when I'm done with it, you're welcome to it." And to that, again, small little town, they don't do what needs to be done to my brain, so I would have to no weeping and wailing in an emergency room if I die. I have to immediately go 50 miles away across the lake to a teaching hospital to get my brain harvested to be able to go to Jacksonville. So my second question is, coming back 1993 to, let's say, probably 5 years ago, when they looked at my dad's slides down in Jacksonville, they said MSA with dysautonomia based on what they saw from his autopsy. I had already had that suggested to me as a diagnosis, so it's not like I'm feeling it because he said it. I heard it first and didn't know what the heck it was, and so everyone says you can't have it familially because it's not genetic. There is some in Japan, but why is my question, I guess I would say. And how do I, who now have 2 children and grandchildren and 3 brothers and 3 sisters, how do I make sure that not only my brain goes, but what do I give them for DNA or anything else to make sure that we aren't creating more of this in our family if maybe it is familiar and they just don't know yet because it's so misdiagnosed or underdiagnosed?
Well, I mean, I think what you raise here is a very interesting issue. I mean, it's not that, you know, we don't have direct Mendelian inheritance, right? It's not like a recessive disease or a dominant disease or something like that. That's not the picture that is emerging, right?
Okay.
But you do hear stories of families that have neurodegenerative diseases in their history. It's not uncommon, whether it's more common or whether it's the same frequency as other diseases, we still don't know. I know at NIH, there's a team, there's a fantastic, brilliant female scientist, Sonia Schultz, that's working on the genetics of MSA. She's collaborating with Lucia Schottlander and Henry Holden, her team in UCL in London. And the idea is like the more samples we can get, the more family histories we can get, the closer we are going to be to tangling out some of these things. I can tell you that as far as we know, we don't think there's a direct passage from mother to son, etc., etc. It doesn't mean to say down the line we may pick up on certain genes that may make neurodegeneration more common for some reasons in other families, but right now we don't have the answer. That's entirely speculation, and that's the best I can do to answer that for you.
Any other questions? That's everything. Thank you, Dr. Norcliffe.
Thank you very
Kaufmann.
much.